Hello and welcome everyone. Previously, we saw that germ theory of diseases was developed by Robert Koch, and then we also saw the basic principles of isolation of microbes were also discovered. In today's class, we'll be seeing the growth of medical microbiology, and these two techniques, that is the isolation of microbes pure culture, as well as the Koch's discovery of Koch's postulates, were also very important in the growth of medical microbiology. Now, during the 1850s to around 1950s, which was known as the golden age of microbiology, we saw wide growth of discovery of microorganisms, the causative agents of different diseases. So, these microorganisms can be bacteria, viruses, protozoa, which we know now. But previously, viruses or bacteria and all this were not so clearly distinguished. So, initially, most of the microorganisms included the main bacterial diseases were first discovered. So, every now and then from 1850s onwards, this discovery started. So, these were the famous discoverers, the organisms they discovered and the diseases that these organisms caused. Now, we also saw that the plant pathology, that is the diseases that are there in plants, were also being discovered, and their causative agents were also being discovered. Like around 1845, the blight disease of potato was identified in a fungus, which is caused by a fungus. Which was caused by a fungus, and the discoverer was A.J. Berkeley, but he wasn't sure that what is the name of the fungus. Later, around 1861 and 1863, the blight disease of potato was caused by Phytophthora infestans was proved by D. Barry. He was known as the founder of experimental plant pathology. Around 1878 also, the fire blight disease of pears was caused by Erwinia amylovora was also proved by Birrell. So, here we see that there is a slow development of plant pathology also. Basically, whatever is happening is nothing but an extension of Koch's postulates only. We were trying to find out that what are the diseases, what are the symptoms, and what are their causative agents. This was happening during 1850s to 1950s, which was called the golden age of microbiology. Next, we also saw the development of virology. Initially, viruses were not known. So, around 1880s, there was a disorder in tobacco plants that the leaves were having a mosaic-like pattern, which was identified by Adolf Mayer. He found that there was mosaic-like pattern on the leaves of the tobacco. Now, what he did was he extracted those infected leaves and extracted that solution from those infected leaves, just like the thing that was told in Koch's postulates, and he injected them in healthy plants. He found that the healthy plants again got the same symptoms as the diseased plants. Now, around 1892, Ivanovsky, he found that the tobacco mosaic disease, which was caused by a pathogen, they had no idea. They had an idea about bacteria only. So, according to Ivanovsky, this bacteria are so small, or these organisms are so small, that they could even pass through Chamberlain filter, which can even trap the smallest of the bacteria. Now, around 1898, M. Bajering, what he did was he found this liquid that can easily pass through even Chamberlain filter, which was a very microscopic stuff, can also easily be trapped in these filters, but these organisms which whatever it was couldn't be trapped and it could easily move out. So initially people thought that maybe it is some kind of fluid which caused the infection. So the fluid was named as poison and it was known as the Contagium vivium fluidum, which is an infectious agent, and he was the first time to call this fluid as virus, which means the poison, and he said that this was the thing that was causing the tobacco mosaic disease. Later in 1935, W.M. Stanley isolated the TMV for the first time and in crystallized form. He also crystallized the viruses. Around 1982, Stanley B. Prusner, he discovered another infectious agent which was not as big as bacteria. It was like viruses, because viruses also had proteins and nucleic acids. Similarly, this was called prions, and they were having infectious agents called proteins, prion proteins. Okay, so PRP. So this will be studying about in details in another topic that is there in this first semester itself. So this is how the development of virology happened. Now we are going to go to the next topic, that is immunization. Now we are going to be seeing how immunization came into the picture. So slowly, as we are seeing, medical microbiology is developing. Immunization became a very important preventive measure. So in this measure, who discovered it? It was discovered by Edward Jenner around in 1798. What he did was he immunized a boy called James Phipps, who was an 8-year-old boy, with cowpox. And when he immunized him with cowpox, he got protection against smallpox. The cowpox virus or anything, whatever it was, which was not known to them. For as of now we know it, but then it was not known. So initially they understood that this particular thing, this cowpox, gave them the protection against smallpox also. This was the first case of vaccination. Later on, even Pasteur found out chicken cholera, anthrax and rabies vaccination also. So as we see, from 1798 to we are going towards 1800s, where the actual discovery was going on. Now, next thing is surgical antisepsis. This was done by Joseph Lister. He proved that initially he was actually seeing that he was a surgeon himself, so he was seeing that post surgery there were some infections. So to stop those infections, what he did was he did a disinfection of the place where the surgery was going on, and also he took some antiseptic measures as well. So around 1867, he said that post-surgery infection is caused due to the microorganisms that were entering the exposed tissues during the surgery. So what he did, he was using disinfectant carbolic acid, which is a kind of a phenol, plus some other antiseptic procedures and measures to contain the infection. So the infection could be stopped by all this antiseptic procedures. So this was done by, first of all, Joseph Lister in 1867. Next we are going to be seeing the rise of chemotherapy. So till now, if you see, what we were doing was we were finding the causative agents of different diseases. We were seeing the immunization procedures. We were seeing surgical antisepsis, where surgical antisepsis could be stopped. All these procedures were all preventive but not curative. As in, till now, people had no idea that chemicals can cure many disorders. Now, Paul Ehrlich started to find out a cure for the different disorders. He found 1,000 new derivatives of a chemical called atoxyl, of an arsenical chemical called atoxyl. He found that the derivative number 66, which was called as salvarsan, can treat a disease called the syphilis, which was caused by bacteria. So, he found out that we can contain the microorganisms by the use of some chemicals. So, this was the first curative discovery, and it was the rise of chemotherapy started from here. So, he called this whole procedure as chemotherapy, where he was using chemicals to contain the microorganisms. Then there was discovery of antibiotics also, which was again a very important substance to cure the diseases. How? It was first seen by Alexander Fleming in 1929 that in his culture of Staphylococcus aureus, you can see this is the culture petri dish. So, in this Staphylococcus aureus culture, we can see the green cultures over here. There was growth of some fungi, which was Penicillium notatum, this red ones. Now, this was a contamination which was coming accidentally in his petri dish. It was not a very, it was an accidental discovery actually. Now, what he found out, that if this is the case, if Penicillium notatum is accidentally coming over here, it won't be a problem. He can make a new petri dish and new culture. That wouldn't be a big deal. But what he observed over here was Penicillium notatum was actually killing the Staphylococcus aureus in this thing. Staphylococcus, which was a bacteria, was killed by the Penicillium notatum fungi. Now, what he did was he grew this Penicillium notatum in a liquid medium. Then he filtered it and he separated the cells from the fluid. Then he found that the fluid, if is again put on many bacterial species, they were again killed by this particular fluid. Their growth was somehow being stopped by this particular fluid. So, it was for the first time discovery of some antibacterial agent, which was now known as the antibiotics. Later, Selman A. Waxman, who was a soil microbiologist around 1940s, discovered that many bacteria of the Actinomycetes group can produce some antibacterial agents which can again stop many growth of many microorganisms, as in the bacteria mainly. Streptomyces gracious was one of that Actinomycetes bacteria which was producing streptomycin, which was an antibiotic. This discovery was also done by Selman A. Waxman and his group. So, this was the overall discovery of the medical microbiology, or rather the development of medical microbiology. So, that's all. Thank you.